Hi, I'm Josh Duggan,
My name's Ryan Fuentes.
and this is our group project on the Forever Fix, gene therapy, and the boy who saved it.
This slide goes over the general contents of our presentation, starting from a base level of the summary of Corey Haas's story, going into more specific things, talking about his disease, the RPE65 gene, the general timeline of his story, the relation to our course content, the demonstration of authentic scientific practices, and finally the references we used.
The Forever Fix covers the story of several patients and families whose journey through gene therapy were resounding successes. However, we are focusing on the crux of the narrative, that is the story of Corey Haas and his fight against Leber congenital amaurosis. The story of Corey Haas is the main crux of the Forever Fix. Corey Haas's story begins with his childhood blindness caused from Leber congenital amaurosis. The disease made him blind from birth all the way until age 7, where we learn of his journey into the field of gene therapy. At age 8, Corey was given the opportunity of a lifetime to cure him of his disease. This cure came from innovations in the field of gene therapy. The therapy treatment given to Corey was through the use of viruses that carried healthy genes which were unaffected by Leber congenital amaurosis. The injection into Corey's left eye had immediate results which drastically improved his vision. The small but resoundingly successful procedure paved the way for not only Corey, but for gene therapy as a whole.
On this slide, we'll discuss the disease that plagued Corey Haas, called Leber congenital amaurosis. This disease is based in hereditary transmission from two carrier parents that are unaffected by the disease themselves. This autosomal recessive pattern of inheritance leaves a slim chance of the carrier parent's offspring to contract the disease at a rate of 25%. Unfortunately, Corey fell into this 25% of contracting Leber congenital amaurosis. The disease itself is characterized and diagnosed by massive loss of vision from a young age. Other symptoms of this disease include high light sensitivity, abnormal movements of the eyes, and the appearance of sunken eyes. The picture included on this slide is an actual photo of a retina that is stricken with Leber congenital amaurosis. This photo illustrates how the eye is damaged from the disease and unable to react to the light being shown through it.
The table displayed on this slide is one that was constructed by us, which references the database used by the online Mendelian Inheritance in Man, which is an organization dedicated to the categorization and compendium building of human genetics, genes, and phenotypes that are present within the human population. This database used by the online Mendelian Inheritance in Man is updated daily and used by scientists around the world. Looking at this table from the top, we see the type of Leber congenital amaurosis, the OMIM serial number, which is the unique digit identifier used within the database to identify genes, the specific coded name of the gene, and the locus, which is the actual position of the gene on the chromosome itself. A closer look at this table shows the specific gene that Corey Haas had been infected by Leber congenital amaurosis, which has been highlighted in red. The RPE65 was the specific gene that was damaged.
For this slide, we will be going into more depth about the actual gene that was affecting Corey's eyesight, the RPE65 gene. The RPE65 gene is located in the retina and is produced in the retinal pigment epithelium. RPE65 helps in the vision cycle. Specifically, it helps with protein production used for normal vision. The vision cycle is the process by which light enters the retina. This light is then converted into electrical signals, which the brain uses for vision. In regards to RPE65, when light enters the eye, it travels to the pigments located in the retina. This light is converted with a form of vitamin A called 11-cis-retinol, which converts the vitamin into a molecule called all-trans-retinol. This whole process converts the light into electrical signals. After this process, RPE65 converts the all-trans-retinol back into the original 11-cis-retinol. The relation of this process for Corey was his original RPE65 gene was almost non-functional. This led to his childhood blindness.
This slide and the next are going to cover the general timeline that is Corey's life and his journey through Leber congenital amaurosis and his success in gene therapy. Corey's parents, Nancy and Ethan Haas, were both carriers for the gene that causes Leber congenital amaurosis. Nancy and Ethan were unaffected by the disease themselves, which unfortunately made Corey a prime target for Leber congenital amaurosis. The Punnett square pictured below puts Corey's parents on both axes. Corey's parents are portrayed with a big H and a little h. The tag pointing out Corey illustrates the disease being autosomal recessive. Being born from two carrier parents resulted in Corey being legally blind up until age 7. Throughout his early life, Corey's parents and babysitter noticed Corey bumping into objects and having difficulties distinguishing features. His parents decided to figure out what was wrong with Corey and took him to the doctors. The doctors tested Corey with an electroretinogram, which is used to check a retina's reaction to light. Unsurprisingly, Corey's retinas didn't react to light.
After Corey was diagnosed with Leber congenital amaurosis, his primary care doctor, Dr. Stephen Rose, pictured to the right, immediately recommended the use of gene therapy to cure Corey's eyes. Although hesitant at first, Corey's parents agreed to the procedure to save Corey's vision. At the time of the procedure, gene therapy was unheard of in the field of ophthalmology for curing Leber congenital amaurosis, which makes this procedure a landmark in the history of gene therapy. The surgery used to cure Corey of his disease was to inject roughly 48 billion viruses into his left eye. The viruses were doctored, which means that they were constructed with two objectives in mind. 1. The genes were designed to leave Corey unharmed. 2. The viruses contain copies of the healthy RPE65 gene to nearly replace all of Corey's affected genes within his eye. After the procedure, Corey was almost able to immediately pass coordination tests as well as other vision exams. This slide covers the demonstrated authentic scientific practices displayed within the book. The procedure that Corey Haas underwent was unheard of at the time in the field of ophthalmology. Using gene therapy to cure Leber congenital amaurosis was a revolutionary procedure that had lasting effects not only in the field of ophthalmology, but the medical field as a whole. As a result of this, numerous medical fields have been researching gene therapy as a cure for many other diseases. To discover Corey's condition, his doctors performed an abundant number of tests such as an ERG, and after discovering Corey's disease, doctors reasoned that gene therapy was the procedure that should be performed to return his eyesight. Dr. Rose and his team displayed ingenious and pioneering procedures that went against the medical grain to cure Corey Haas.
This slide will cover how all of this content relates to the course. First off, with genes being such a large subject matter within the book, there was a strong relation between the genetic section of the class and what we read. Specifically, the RPE65 gene is an examinable gene itself with details such as its gene identifier and locus. In class, we discovered the importance of how even a single gene can greatly affect a person's life. Secondly, the use of gene therapy covered by Emily was brought heavily to light. In Corey's case, gene therapy was able to restore not only his vision, but his ability to live a normal lifestyle. The gene therapy subject related to class as we learned how the manipulation of genes can be used to cure disease. Finally, the overarching theme of genetics was important not only to the book, but to the material we covered in class as well. We learned how genes were passed on from parent to offspring in numerous ways. In this book, Corey's autosomal recessive disorder was quickly identified by us as a genetic defect. These are the references that were covered and used within this presentation. Thank you for listening to us. The content we covered in our biology course and the knowledge we learned from the Forever Fix and gene therapy as a whole made us greatly appreciate and delve into the world of science.